incident was about to take place that would shake the foundations of the civilized world.
It is time to open your mind. Time to discover your divine self as we navigate this holographic matrix together. Strap in, cosmic family, as we slide through this wormhole of cosmic information together in search of the truth.
You can't handle the truth.
It's time to go inside the matrix. With your host, Jimmy Brent.
All right, welcome everybody to Inside the Matrix. I hope that all of you are well out there and managing to survive and thrive as best you can in our crazy circumstances we find ourselves in. And before we get started with the show, I just wanted to take a minute to thank everybody who contributed to the show. Last week we got some contributions to help us keep the show going because, as you know, so many of us are out of work and just trying to keep our households going. So I greatly appreciate the contributions. And if anybody is still willing to help out with whatever they can do, it's always appreciated. And there is a donate button on the kcorradio.com homepage. Anyway, I really hate even having to say that, but I just wanted to at least thank all of you who did help out. I appreciate it immensely. So our guest today is Dr. Andrew Kaufman, MD. Dr. Kaufman has been making the rounds on a lot of YouTube shows and a lot of the best shows around of the so-called alt news media, but I think we probably refer to it as the real news media these days. And he's a super intelligent guy, and he's very astute on finding the best scientific articles which help shed light on what is happening in our world today. So Dr. Kaufman, may I call you Andy? Welcome to the show.
Thank you very much, Jimmy. Yes, please call me Andy. I don't really like the title, to be honest with you, but I know it gives some credibility, so it is important.
Yeah, absolutely. And, thank you for being here. And I've heard you on a bunch of different shows, and the information that you have is so important for people to understand at this time, because there is a major social engineering operation going on right now, and we can get into that aspect too. But let's start off with, if you wouldn't mind, I usually ask my guests to kind of give us a background on sort of how you grew up, what got you to where you are today, just in a brief nutshell of your life, so people can get an understanding of where you sort of came from.
Sure thing. Well, yes, that's a great opportunity. So I grew up in New York City, in Staten Island, for the most part. My father was a computer programmer. My mom was a high school teacher. And so education was always emphasized in my family. And ever since I was very young, I was very interested in science and how the world works, and especially the human body. And I used to, every time I had to go to the doctor, I was always asking lots of questions, and I was really attracted to being able to, help people get better and things like that. So that really stuck with me. And then when I went to college at MIT, I thought about a lot of different things. I studied some electrical engineering before I realized that wasn't really my thing, and ended up basically deciding that, medicine is the right path for me. And I took a little bit of a long path because I spent some time doing research and exploring other careers. So I worked in the biotech industry and helped start a molecular modeling department, which is a way of using computer simulations to discover new medications. And I supported a large manufacturing facility subsequently to that, and even worked for a CDC-sponsored position for the New York City Health Department, doing surveillance for AIDS cases. So I would go around to hospitals and clinics and identify who met the criteria for AIDS. And this was in the early 90s, when things were much more prevalent than they are today. And eventually I decided to go back to medical school. And I did that in Charleston, South Carolina, which is quite a nice place to live. And it was a very good school because since it wasn't one of the like upper echelon medical schools, they really focused on clinical medicine, which was great for me to learn at that time. I did participate in some research projects, but I really needed that opportunity to hone my skills on how to work with people. And it was great. And then I went to Duke, a very different kind of institution after that, to train in psychiatry. And there, that is a heavily influenced research facility. So I did get involved in several projects and had a couple of early publications. And during that experience, I heard of this subspecialty of psychiatry called forensic psychiatry. And, it's something that most people don't even know about. These days, they may recognize it from some TV shows like Law and Order, but they don't really portray what it's like in the real world, of course, because it is TV. And but the interface of law and medicine and law and psychiatry was just fascinating. And there's was an elective where I could actually study some of the case law, like a lot of Supreme Court and other appellate court decisions related to the rights of people with mental illness or related to the insanity defense for like a murder or other serious crimes. And it was just fascinating. And I've always been a person who likes to debate and discuss things. And that seemed to give me some kind of a forum for public speaking and making a logical argument, and the challenge of being cross-examined was, kind of exciting. So I decided to pursue a fellowship in that subspecialty and came up to Syracuse, New York, where the director of the program, James Knoll, was someone I really looked up to. He had accomplished many great things in his career, and I remember that I went to a conference either before I went to the fellowship for the professional organization. And this is the first time I had the opportunity to meet Dr. Knoll. I had just known of his work prior to that. And he was giving a panel about Jim Jones' cult. This is, if you'll remember, the cult where everyone took the Kool-Aid and suicided. Mm-hmm. And he actually had one of the very few survivors from that cult talk at this panel discussion.
Wow.
And it was extremely moving to hear this man's story because his own family, he couldn't stop them from drinking the Kool-Aid, even though he tried desperately. And he really laid out how Jim Jones was able to trick people into thinking that he had special knowledge or was ordained by God in some way, such that they would join up the cult. And of course, they targeted very vulnerable people who were marginalized and alone, and that's how the game works.
Yep.
But he gave a very inside view to this, and actually Jim Jones was really struggling himself, which is why he came up with the ultimate plan that he was a serious drug addict and he was really losing control of everything. So that was such a moving experience and, significant in my life and perhaps even relevant to what's going on today with all of the misinformation and manipulation of people's emotions.
Absolutely.
So after I completed the fellowship, and that's when I really increased my effort into research, and I learned how to do statistics and even program my own statistics in the software, which is very uncommon for physicians. Usually they pay someone or they have someone at the university, like a statistician who does that. But I think it's very important to be able to analyze your own information and data so that you fully understand the repercussions of what you're doing to it and how it might create biases or influences. So this was a really important lesson for me, and especially when I look at scientific publications and medical publications, that I use a very discerning eye. And I'll tell you, one of the most important things is really to make sure that you always read the design of the experiment. They call this the methods section of a paper, because that tells you if they actually did the right experiment to prove what they're saying they proved. And if you don't pay close attention to that, you could end up coming to the wrong conclusion about what their data is showing. And that is very relevant in my recent research. So I joined the faculty after that, and the first thing I got involved with was being the research director for the fellowship program. So I began mentoring the new fellows and supervising their research along with a partner who is a PhD. And that was a very interesting and rewarding experience. And as I stayed in the department, I was earned several leadership positions in the Department of Psychiatry there, including the vice president of our faculty corporation, which was a little bit more of a business role, but a lot of management of the personnel. And while I was doing that, I got this idea for a way to use a medical device to prevent suicides that were occurring in jails. In jails, actually, the rate of suicide is several times higher than in the general population because of the, severe stress that people are under while they're going through a criminal proceeding, as well as the extremely ascetic and difficult conditions inside of a jail.
Yep.
And so I took some time where I decreased my faculty responsibilities, but I still did half-time clinical work for the university while I had a full, more like a full and a half time job trying to run a startup company for medical devices. And boy, I'll tell you, that was really a learning experience, because when you're trying to start up a new company, what you'll find is that every system out there is really against you, especially if you're developing technology, that there's just roadblocks everywhere. Like, for example, I was awarded this grant from the state that was a business development grant, and they didn't really give any guidance or instructions, but I learned later that it was actually taxable. So I got money from the government, and then they wanted a third of it back.
So that's not really a grant, is it?
No, it's I don't know what it is exactly.
It's like income.
Right. And then, that's just one of many, many examples of how difficult it is to get things off the ground. And I remember at the time, I had a patent application in, and I felt that it was actually quite strong because I had discovered something that was very unique that was not in any of the other patents in this space. However, all of the so-called experts who were investors or attorneys, or I even had a class at the law school in patent law where they analyzed my company's intellectual property, and everyone said, oh, you'll never get a patent, you'll never get a patent. And that is worth value, because any investor in a company, they want something tangible. So they need to believe, of course, in the person, but the intellectual property, even if the person fails and the business fails, that still can be sold on the secondary market. So if they invest in that, then it's much less risky. And of course, I got to a point where I had several partnerships with some big companies to help me develop the prototype. And they just, at the last minute, their whole department said they got their funding pulled, and this was Philips Electronics.
Wow.
And then, I had so much invested, and almost all of the grant money was gone. And then my consultant, who was actually the engineer who was going to build the prototype with them, said, I'm tired of waiting, and he quit. And so I'm left with no technology, no engineer, and no money.
Oh, nice. Yeah.
And of course, 2 years later, I get a notice from the United States Patent and Trademark Office that the patent was granted.
Oh, my gosh.
So too little, too late. But, live and learn. Got to chalk this up to life experience. And to be honest with you, I didn't really want to be in business, running a business. I'm much more of a guy that wants to try and help people and get the word out there about important things. So in a way, it's kind of a good thing that it ended that way.
Yeah, and I know a lot of people probably don't realize that the oversight of a lot of the patent offices is farmed out to private corporations, and one of them being a corporation on the Queen's Privy Council. And although they are incorporated in the U.S. as well, their parent company is in the U.K. So a lot of patents are actually stolen and then weaponized, if they can be, and then just taken away from people without, and then their patents usually get denied after that. Or if they do get it, somebody else is already utilizing it. So that's fairly common thing. So when people think that they're going to be protected with a patent, if there's any way that patent can be weaponized in some way, whether it's software or hardware, it generally will be one way or another. And it's a sad thing, but that's quite true. It's amazing to me, most medical doctors, whether they're in psychiatry or general practice or specialists of some kind, I think a lot of them are so busy with their actual practice, they don't get a chance to jump into the more scientific aspects of it. And I believe you mentioned on one of the shows that a lot of the doctors just read the sort of summary of a lot of these studies, and they don't actually look at the studies to show what the actual conclusions were outside of the summary, the more detailed conclusions. You want to talk about that a little bit?
Yeah, absolutely. And I think actually, and I did talk about this a little bit, but I want to give you a little bit more detail because. Yeah,
and I'm sorry, didn't mean to interrupt your
oh, no, not at all.
There.
Not at all. Believe me, this stuff is much more important than my personal history. So when I was in medical school, they were starting to talk about the importance of learning how to read articles and even do a few statistical calculations, like things like sensitivity and specificity, which means, let's say you have a diagnostic test that's a screening test. Like, let's say, a test for cancer, like prostate-specific antigen or PSA. I think most people are familiar with that, at least middle-aged men are. Mm-hmm. And so if you want that screening test to pick up every possible case of prostate cancer, you want to make it very sensitive. But of course, when it's very sensitive, it's going to create some false positives. So you have to have like a confirmatory test. And so they wanted us to actually be able to look at some data and be able to calculate the sensitivity, which would be expressed as a percentage. And this was like a new course, and they wanted us to pass an exam to demonstrate some basic competency in doing this. And this caused quite a kerfuffle among the students because they are kind of afraid of math. I think a lot of doctors come to medicine from liberal arts backgrounds or other backgrounds. They really like to have well-rounded people. So they're not really scientific, technical kind of people. And then when they're asked to do some statistics, they get a bit shy. And so this was like the class that people failed. And then they had to remediate it. So just to give you an idea of what, the general average doctor might be like. But most doctors, as soon as they finish their training, there's a lot of delayed gratification through your medical training. It's a very long training, 4 years of medical school after your college degree, and then, up to 9 or even 10 or 11 years of residency training, depending on what specialty you go into. My training was a total of 5 years. So while you're in school, you're unable to work or earn income, so you're basically living on, handouts from your family or loans. And so you're generating a bunch of debt for that 4 years. And then when you start doing your training, which is just like an apprenticeship, they pay you pretty darn low wages, especially if you consider the amount of hours that you're working. So if you whittled it down to an hour rate, it was pretty pathetic for the amount of training and qualifications you had to have and how hard they made you work. Mm-hmm. So, with that kind of income, you could definitely, afford to buy a very modest home or pay your rent and your food and stuff with your income, but there's not any left over for anything. In fact, I didn't know any doctors who were really able to even start paying back their student loan debt at that time. So you're kind of living, at a reasonable standard of life, but not what you expect, not really being able to pay back your debt. And so as soon as you get out, you can't wait to, buy a house, get a car, send your kids to private school. And so then you accrue more debt, of course, and you become somewhat of a slave to that because it's always there, and then you have to work hard enough and get a high enough paying job in order to pay it back, and then still have enough money to pay your own expenses, and perhaps you feel like you deserve some luxury items or nice vacations at that point, so people overextend themselves. And they get into this situation where they realize that it's not quite as lucrative as you might have thought it was, and you really have to work your butt off to make ends meet. And so the insurance industry and managed care have really been responsible for this because they tell you how much they're going to pay you for a service.
Right.
It's a strange industry because, I mean, what other business do they have any business telling you what to charge for what? I mean, generally, it's the market forces that determine the pricing. But there's none of that in medicine. It's all regulated to some degree behind the scenes that you just basically have to accept it if you want patients to come to you. And so they want to pay you a very low amount per patient. And that's really important because in order to spend the appropriate time to really talk to somebody and to do a proper examination to know what's really going on, you can't do that in 4 minutes. No matter how efficient or bright you are, it's just, people can't explain themselves in that time. You need to have a back and forth in order to get at the right information to know what's really going on. Right. But that, if you did it that way in any setting where insurance is paying, then you would basically go bankrupt in a matter of months. So there's this pressure to see as many patients an hour as possible. And in psychiatry, this was really an issue because psychiatry is all about talking. And you have to get someone talking in order to know what they are really experiencing. And
of course, yeah.
And there are some practices where they will see 10 patients an hour, so 6 minutes.
Oh, my God.
So it's basically long enough to write a prescription.
Well, this system is so obviously completely broken. The insurance companies being the middlemen and, like you said, dictating, how everybody's going to operate. This is totally insane, and that this is not really capitalism at all. It's just a system of control. And of course, the insurance companies are hand in hand with the pharmaceutical companies, and they are very much in the middle of all this too, controlling things and rewarding or punishing doctors for their participation in writing these prescriptions and all that. So, we know that's it. The system is completely broken.
Well, I'm not sure what happens behind the scenes, but actually the pharmaceutical industry and the insurance industry are a little bit at odds because the drug companies want the insurance companies to spend a lot of money on the drugs, and the insurance companies don't want to spend money on anything except denying services. So it's quite a complicated relationship, but for the doctor, it's a real conflict of interest because if you're getting paid by the insurance company, then who are you working for?
Well, you're also getting paid, incentives by the pharmaceutical companies too, right?
Well, only if you want to willingly participate in that. And that's something that I think most physicians stay away from, but certainly some really take advantage.
Yeah, for sure. All right. Well, we're going to take a short pause right here. We're coming to the end of our first segment with Dr. Kaufman, and we're going to get into a lot more about what's currently happening in this next segment. So everybody stick around with us. If you have a phone call, please give us a buzz. If you want to put something in the chat, if you have a question, feel free to do so. We'll be right back. Okay, welcome back, everybody, to Inside the Matrix with our special guest today, Dr. Andrew Kaufman. One thing that I think a lot of people don't really understand is this whole concept of germ theory. Now, they understand it from the standpoint that it's, people talk about how you can catch this and catch that, and that's the whole point of the crisis that we're being exposed to right now. And this originated, I believe, with Louis Pasteur and his work. which was, I believe, later refuted even by him. But Andy, would you mind going through the whole thing about germ theory, what it means, what it doesn't mean? Is it valid? Is it not valid? All those things. Can you give us your insight on that?
Yeah, sure. I'd be happy to, Jimmy. And actually, it dates back a little bit further than Louis Pasteur. And Louis Pasteur was an interesting gentleman because he was actually trained as a chemist. He was not a doctor or a biologist. And much of his work was actually not original, and much of it was plagiarized or copied from some of his contemporaries who were much more serious scientists.
Sounds familiar, doesn't it?
Yes,
Nikola
it does.
Tesla. Yeah.
And
go ahead.
actually, one of those serious scientists was by the name of Antoine Béchamp. And this was a gentleman who basically his only goal was to learn about the world. And the main experiments that Pasteur alleged proved germ theory were actually repeating the same experiments that Béchamp had already carried out. And I'll tell you what's really fascinating about this is that the experiments were about fermentation. So I think your viewers will recognize that fermentation is how you make beer and wine. So you have some plant material, and there are some. Right. Yes. Sauerkraut, all the fermented foods that are gaining popularity that we used to eat a long time ago, and they can be very healthful. And so they are basically comparing fermentation to illness in an animal host, even though they are quite different processes. And fermentation actually really in nature exists to recycle and break down dead material. So you would have, dead plant material, and microorganisms may come from inside that plant from when it was alive, and may come from the air. And there were experiments proving both of those performed by Béchamp. And it basically decays the dead plant material so that it's broken down into the elements and can be reincorporated into new life. And this is how, all the nutrients get back into the soil. And we've all seen lots of examples of this in our life, walking through the woods when we see decaying plant matter, or even a dead animal body, which a very similar process occurs. It's not called fermentation in that case. It's referred to as putrefaction. And during that process, as we know, that a lot of toxic chemicals can be created by the microbes, which make it very foul smelling. And we know that if we, touch dead bodies and then put our hands to our mouth, we can get sick from those poisons. Mm-hmm. So in actuality, the original germ theory evidence comes from fermentation, not from any disease in people. And Pasteur, as you said, had this colleague, Claude Bernard. And Bernard originally was a proponent of the germ theory, but he did additional experiments on his own that disproved it. So, in other words, showed that fermentation can occur when there's no outside microbial agent. And Béchamp had also performed similar experiments. So, in other words, the infection can occur without any outside microorganism invading the body, which is very different from the germ theory of infection. Right. And Claude Bernard had talked to Pasteur about this when he was alive, and Pasteur told him, you better keep that to yourself, because he knew it would ruin his reputation and his livelihood. But on Pasteur's deathbed, and I looked up the references, and it's not 100% certified that he said this, but he's widely reported to have made this quote that Bernard was right, and the germ is nothing, the terrain is everything, was the statement. And so if you look at the science that came after that, you'll see that there is really no experimental evidence that backs up the germ theory. And that's why it's still called germ theory, because a theory is an idea or a hypothesis, which is kind of like a guess to explain something that you observe. But when it's proven, then it becomes a law. Like we may remember from high school chemistry and physics, the universal gas law. And if you apply those equations to a system of gases, it's going to come out exactly right every time. So that was proven experimentally, and then it became a law. But germ theory never became germ law. It stayed germ theory because it's never been proven. So subsequently, as germ theory developed, this German scientist came along, Robert Koch, and he's one of the big shots in germ theory. In fact, in Germany today, they have a scientific institute named after him where they still do this type of research. And Koch came up with these 4 postulates, which was essentially a set of rules by which you could design an experiment to prove that some kind of microbe, infectious microbe, could cause the disease. And they're pretty straightforward. I mean, if you just think about it rationally, you would come up with this on your own, because basically you take a sick person and you're able to take out some of the part of their body that's sick. Like, let's say you have a strep throat, right? So you would put a cotton swab in the back of the throat, and then you would be able to take out the streptococcus bacteria from that and isolate it and culture it in the laboratory and show that it's there. And it's important to also note that you would have to also have a healthy person and do the same kind of test and demonstrate that that particular microbe is not present. Because if it's present in healthy people and unhealthy people, then it can't cause the disease, because how would the people be healthy? Right. And then you would take that purified microbe and put it into a healthy person, and then it would show that they would get the same disease. And then Koch suggested actually that you repeat that from that person one more time, that you are able to isolate it back from the healthy person that you just made sick, and show that it was still there. Okay. Mm-hmm. So if you look, though, for experimental evidence for any particular disease that is supposedly caused by a germ, you cannot find these experiments that were successfully performed. And the reason that I brought up strep throat specifically is because streptococcus is actually a bacteria that is in all of our bodies. And usually you could actually culture it from your throat. We certainly know that it's on the skin and in the digestive tract, but often in the mouth as well. So if your doctor started doing strep throat tests when you didn't have any symptoms, they would be positive most of the time, just like they're positive most of the time when you have a sore throat. But since the streptococcus bacteria is present in sick people and healthy people, then it can't possibly be the cause of the sore throat illness.
Makes sense.
Yeah, it's pretty straightforward. So I have not exhaustively searched for every single infectious disease to satisfy these postulates, because after I looked up a few and determined that it has not been satisfied, in my mind, there was no reason to look further. So instead, I looked to some other researchers that have done that work. For example, Dawn Lester and David Parker, who have an excellent book that has been released.
I have it.
So it's called What Really Makes You Ill. And I've actually interviewed them on my show as well. They're a wealth of knowledge. And they looked much more exhaustively than I did. And not only could they not find evidence of satisfying Koch's postulates for every bacterial and viral illness that has been purported to be infectious caused by a germ, they actually were able to find considerable evidence of alternative causes of many of the illnesses that were thought to be infectious. So this was quite a revelation.
Absolutely. It's almost 800 pages, this book. This is a huge reference book, and I highly recommend it to anybody who wants to get it.
So there is a contravening theory that has also been developed, because, the reason that they got some of these ideas when they started to look under the microscope, because, none of this was possible before the microscope was invented by Anton von Leeuwenhoek, because they didn't know that microbial organisms or microorganisms existed, because they were unable to be visualized. So they had theories about, various things that could cause disease or that were in the air, but they had no proof. So, at some point they were able to visualize on our tissues that these microbes were present, and they were present when there was disease. But this is a incidental finding. So it would be the same thing as, let's say that you were observing neighborhoods, and you came upon a house that was on fire, and there were firefighters there putting it out. But since the firefighters were present, you would assume that they caused the fire. And obviously that's a pretty ridiculous example, but that's exactly how germ theory was formulated. They happened to see the bacteria there, and they assumed that they must cause the disease.
Yeah.
But remember what I said earlier about how we've all observed in nature what happens with decaying plant and animal matter, that these microorganisms break it down for recycling purposes. And that's exactly what happens inside of our bodies when we have these illnesses that we're told are an infection.
All right, so let's get into defining some of these microbes that we're talking about. What is a virus? What is a germ? What are these things? Actually, now we know what the scare tactics have told us, and we know that this incredible social engineering action that's going on now has been telling us, but what are these things?
Yeah, sure. Well, I think I want to start with the word germ, because we all have grown up thinking that germs, we equate them with microorganisms, and we see them as something evil that will invade our body and make us sick. Mm-hmm. But actually, the word germ refers to a budding or a new growth. And in biology, the germ cell is the egg and the sperm. So they're the cells that create life. So thinking about a germ in terms of germ theory actually completely inverts the meaning of the word. So going from something good that brings new life to something bad that brings illness and death. So whenever I notice that a word meaning is inverted like that, it always makes me want to investigate how that came about and why it is. So let's talk about viruses, because there's another word that now this meaning has not been inverted, but the meaning actually is from the Latin meaning a noxious substance or poison. And that is the theory about what a virus was before microbiology. So they actually used the term virus in medical research as a cause of disease before they even knew that any microorganisms exist. And what they were talking about is that a poison enters the body and causes illness. And of course, there are lots of examples of how poisons and toxins cause illness. We all know about things like mercury or arsenic. We know about pesticides and herbicides and other organic chemicals that are toxic. So this is compatible with the original definition of virus and the thinking at the time before microbiology. But early in microbiology, they just had optical microscopes that, just use glass lenses, and that technology only allows a certain amount of magnification. And you can definitely see bacteria cells, which are single cell organisms, and you can see fungi like molds, for example. But as they rolled out, bacteria as the cause of disease, because what happened is someone would be ill with a certain disease, and then they would look under the microscope at that part of their body, and they would see bacteria. So they would say, okay, this bacteria that we see in this disease causes it, even though it was just there like the firefighters.
Exactly.
And as they were going through diseases, some diseases didn't really have bacteria there that they could blame it on. So they theorized that there was something else there that was invisible to them at the time, and they called that a virus as well. But they didn't really know what it was. And it wasn't until the electron microscope was invented, which is a much more powerful way of magnifying images, but it doesn't work on optics. It works by a different principle. And then they were able to see that there were, in fact, particles that were much smaller than cells that were present in some people that were ill. But they didn't really know what these particles were at the time, but they blamed them for the illness nonetheless. And so in order to show that the virus would cause an illness, you have to go through the same procedure that I discussed before with bacteria, according to Koch's postulates. So you would basically take the ill person and out of the sick part of their body, isolate these particles. And when I say isolate, I want to specify that a lot, because that word has actually been misused in much of the literature. So in my opinion, the isolation actually means to separate it from everything else.
Yeah.
But when they say this in the literature, they don't always mean that. They mean isolate it out of the body along with some of the body's cells. And this is why you see almost all of the pictures of electron microscope images of alleged viruses are actually they're inside of some kind of host cell, like from an animal or a human. And that's not isolated. That is combined with another type of organism,
Mm-hmm.
if you want to call it an organism. So they've never really, for any viral illness, not even close, demonstrated that they could isolate and purify these particles out of a sick person and not out of a healthy person. And then they would have to do additional steps, but they've actually never satisfied that first step. So there's really no conclusive evidence at all that a virus causes a disease. Now, they have been able to purify some viruses, especially out of single-celled organisms. So there are particles that they call viruses in bacteria, for example, and also in other single-celled organisms like algae and amoeba. And these have been observed under the electron microscope, and they have been able to do protocols where they do isolate and purify these so-called viral particles. And so they can at least prove that they exist. And they have done further experiments where they've studied the structure of these particles. And if you look at pictures of these, they look quite distinct, actually. They have very complicated geometric forms that every single particle looks identical. And they usually have like this head and tail structure, which somehow allows them to get in and out of the cells easily, and so that they can do some of their functioning. And inside of these geometric structures, which could be made of a variety of materials, mostly proteins, they contain some small piece of genetic material themselves. Now, not enough genetic material with genes to be an independent life form where they could reproduce on their own or ingest nutrients and excrete waste products, like all of those general life functions. But
in other words, it's not alive.
Exactly. It is not alive. It is not capable of existing or performing any life functions at all on its own. And it needs to go inside of a cell and basically become part of the cell's machinery in order to propagate in any way. So this is definitely not a life form. It's an inert type of particle. And so they have characterized some things about these viruses, but very different from all of the images of viruses that are alleged to cause illness in animals and humans, where they have various different morphologies. They look more just like sacs or vesicles, not with this complex geographic structure. So it seems to me that they are in many ways distinct entities from what you see in these lower organisms. But I think they're, have not been fully well understood. until recent years, when there's some interesting evidence that is leading many of the scientists to have a better understanding.
All right. So now that we're saying that viruses do not live, they're not living things, and when people in the media and other supposed scientists come out and say, oh, it could live for 5 days on a piece of metal, or it could be, pass from one human to another, and you better get your mask on and all that stuff. What does that mean?
Yeah. So what that has nothing to do with being alive or not alive. What that has to do with basically, is it still intact as a particle, or has it essentially just decomposed into dust? Okay,
so what are we seeing with this panic? I call it a plandemic. You can tell what I'm thinking about it, but what are we actually seeing here, in your opinion?
Well, what we're seeing, in my opinion, is quite a lot of obfuscation. I've looked at the original and subsequent papers where they claim to have isolated and shown this novel, what they call a coronavirus, to cause this illness. And if you look at the experiments that they've done, they have not proven this at all. So what they have done is they've taken just a very small number of sick people. In the first study out of Hong Kong, there were only 7 people, and they actually only had success in 5 out of the 7. So they couldn't even do all 7 of 7. And what they did is they took fluid out of the lungs and they assessed it for genetic material. So RNA, which is a type of genetic material, there's DNA and RNA, and they sequenced that material and compared it to other coronaviruses that they did virtually the same type of experiment with, and said, well, it's pretty similar, so therefore coronavirus caused this illness. They did not try to do the same procedure in healthy people to show that it was not present, and they did not isolate and purify any particles and show them to actually contain the DNA that they were testing. Instead, they mixed it with lung cancer cells, and we know that lung cancer cells actually send out their own particles, which, when I later looked under a microscope are essentially look identical to what the pictures claiming to be the virus are.
So we're coming up against a break here, Andy, and what we're seeing really is nothing scientific at all, but it's a sort of a planned hysteria over this. We're going to get into this a little bit deeper on Inside the Matrix, so if you have any questions, pop them in the chat or give us a call. We'll be back in just a little bit. Stick around with us. All right, welcome back, everybody, to Inside the Matrix with our special guest, Dr. Andrew Kaufman. Now, a point that I want to make is, there are lots of scientific papers backing up what Andy's saying here. This is not just his own speculation or anything like that. He's a scientist as well as a medical doctor, and there are a bunch of others out there who have been quite popular on YouTube and on other social media, such as Dr. Robert O. Young, Dr. Thomas Cowan. There's a gentleman from Australia named Tom Barnett, and there's others too. But many of these videos on YouTube have been taken down and also deleted from Facebook, because this flies directly in the face of the narrative that we're being exposed to with this mind control program that we have going on right now. So go ahead and continue, Andy, with what you were talking about, and what this actually is. Now, this so-called coronavirus, and you were mentioning that the images that we're seeing are not really of a virus at all, right?
Well, the term virus can get very confusing here. So what I really am saying is that they are not what they seem to be or what we are told they are. So in other words, if you took the lung fluid out of a sick individual with a new illness, and you could isolate and purify a particle right from that fluid, not mixed with anything else, completely pure. Then you could characterize it, so you could identify what the chemical constituents are of it, and then you could see if it contains genetic material, and then you can describe that. You can take the sequence of the genetic material to know that it came from that particle, and then you could take that particle and put it in a healthy individual, and it would have to cause the same illness as the person had that you took it out of. And so nothing close to that has been done to this point, and that's really the only way to actually show there is a particle there, and then you could call it a virus if you wanted to. But the procedure that they did, and there were actually two different studies that both claimed that they did what I just described, but neither of them did. And what they both actually did was they took fluid out and they got RNA from it, and they sequenced the RNA. But they don't know what the origin of the RNA was because they didn't purify a particle and then take it out of the particle.
Right.
They just took it out of the fluid. So there are many, many sources of RNA that would be in our lung fluid, not just from our own cells, but from any organisms we may have inhaled in the few minutes before the sample was taken, from our own bacteria and fungi that live in our body normally, and they would be more likely to be present when there is some kind of lung illness because they're coming to perform their recycling role. So it's really, there are many, many sources that this genetic material could have come from according to these experiments. But what they then further did, and this is where they claimed they isolated a virus, is that they mixed that lung fluid with a cell culture. So in the first study, they mixed it with lung cancer cells. Now, lung cancer cells have been studied quite a bit, and they're very familiar to laboratory scientists. And lung cancer cells, as practically all cancer cells, are known to put out particles that contain genetic material. And these particles are referred to by the scientific community as exosomes. Exo meaning outside of the cell, some meaning body. So a body that goes outside of the cell. Pretty simple description.
Right.
And these exosomes are not 100% known all the details of their role, but they are widely reported to be involved in two processes. One is what's called intracellular, so between cells communication, because cells that are right next to each other can communicate directly, but if they want to communicate information to cells that are far away, this is one way they can do it. They send out exosomes. The exosomes contain information, which is in the form of genetic material, and they go and find another cell to communicate that information. And they have this special, they call it a receptor on them, and it's basically like a key. And the key only fits a particular kind of lock. And so they get put into the fluid outside the cell and go into the blood and move around the body. And eventually they find where their key fits the right lock. And that is called the target cell. So that's the cell that they're supposed to communicate the information to. Right. So this is a very important role. And then they've also been shown experimentally to perform another role, which is to remove poisons from the body. So there was a great experiment they did where they had a bacterial colony that was making these toxic substances called endotoxins. And these toxins will actually, it's not the bacteria attacking, it's the toxin attacking a cell and makes little holes in the cell membrane, and it leaks out all its contents like a burst water balloon, and then the cell would die. And so they had cells that had no exosomes. So our cells make exosomes when they're under some kind of stress. Like it could be, there's poisons of various sorts, like heavy metals or pesticides or industrial chemicals.
Or radiation.
Yeah, well, that's the next thing. So ionizing radiation, like X-rays and gamma rays, things like that, from psychological stress, such as extreme fear or panic, from asthma, things like that, infections. So virtually any acute illness or acute stress situation can actually cause cells to release exosomes. So they did this experiment with some cells that were under stress and releasing exosomes, and those cells, when they mixed with the bacterial toxins, the exosomes actually gobbled up the toxins, and they have demonstrated this under an electron microscope, so you could see very clearly. And so this would be a sort of a detoxification pathway. And these have not been identified specifically as associated with the immune system, but clearly they play the same kind of role that the immune system plays. So they probably have some communication, and they have been demonstrated to have those receptors that are specific for immune cells. And those are called MHC, and it's actually the same receptor that they use for tissue typing for organ transplants. So, and that's, to protect against rejection. So we know that the exosomes can signal the immune system, and we know that they can signal other parts of the body, and we know that they can gobble up toxic materials. And when they mixed cells without exosomes with those endotoxins, those cells died. But the cells that had the exosomes did not die. So this shows that they have quite a protective function. Mm-hmm. So basically, you have something that causes an illness, and as a response to that illness, in order to alleviate it, that's where the exosomes come from. So in the experiment where they said that they isolated a virus, what they actually did was they just isolated the exosomes from the lung cancer cells, and they called that a virus. And so I wanted to characterize that a little further, so I did some more research because they describe certain things about these particles. Like, for example, they described the specific receptor, and it was called an ACE2 receptor, or angiotensin converting enzyme. And that's, most of us might be familiar with that enzyme from a certain type of blood pressure medication called ACE inhibitors that block the activity of that enzyme. But you find that enzyme on the surface of several cell types around the body, and that's basically where the exosomes would be targeted. So they said this ACE2 receptor was on the virus particles, and then I found studies that showed that exosomes also have the same exact receptor. And I thought this was just too much of a coincidence because the experimental procedure clearly provided a source of exosomes. Exosomes have the same properties. They have the same size. They look the same. They have the same structure under the microscope. They both have RNA rather than DNA or any other kind of genetic material. And they both were demonstrated to have an ACE2 receptor. So what it seems like is they actually isolated an exosome and then called it COVID-19, but it was no such thing.
All right. So, now they've created all this hysteria around it. And, my personal speculation on this is that there's a lot more to this whole story, and there's other things getting on. And maybe we can talk a little bit about that later. But one thing I wanted to address with you in that, I'm constantly ranting on my Facebook page, and people write back and, oh, haha, you think germ theory is not right, huh? Well, how come when somebody in the neighborhood gets chicken pox, everybody I don't know if they still do, but they used to send their kids out to play with that kid so that they would get it too and then have the immunity. So how do you feel about that?
Well, I think this is the thing that is really tripping people up to try and understand what's going on with this virus.
Right.
And I want to say that, the most important thing is that you have to demonstrate actually that there is a virus and it causes the illness. Because if there is no virus that causes the illness, it doesn't matter how it may or may not get transmitted between people, because there's nothing to transmit, because they haven't demonstrated that a particular thing causes the illness. So even though we have lived our whole lives and we've seen what we think is examples of contagion or people passing disease back and forth, we really don't know that for sure, because we haven't proven that there is a specific agent that causes that disease. So if we reason based on an assumption that's a lie, then it doesn't really matter what the truth is about that thing, we have to go back to the assumption. And we have to say, well, we have to go back to the drawing board and find out what actually is causing this illness. And then we could probably figure out if there really is contagion passing the illness back and forth between people. And I'll give you an example of this that all of your listeners will be quite familiar with because we've all had some experience like this. So let's say that you go out with a group of people for a restaurant meal together. Right? Let's say, there's 20 people and you all live at different places. And, this is a family thing. So it's a group of families, so maybe 4 or 5 people in each family. And so everybody has a great time and they go home. And then in the middle of the night, in various of those households, there's people waking up and vomiting and having diarrhea. Right? And in each family of 5, maybe there's 2 or 3 people who are sick and 1 or 2 who are not, right? And this is basically the experience that we have with, colds and the flu, that we know that it seems to happen that maybe often more than one person in our household is sick at or around the same time. Sure. But not everyone. Right. It's very rare that every single member of the household. And so when, you have all these people under the same roof in each other's personal space, if you could really pass it so easily, like wouldn't every single person get sick? But that's not what we experience. So let's go back to the example where people are throwing up and having diarrhea. Well, what was the cause of that? Did they pass a virus back and forth between each other? Or did they all happen to have the one dish that was poisoned, the dish with the food poisoning? Maybe I said it was the fish when I was describing this one other time, but I was criticized that I had something against fish. So let's say this
you don't
time
want to discriminate,
right? So let's say it was the chicken. There was chicken that was left around too long before it was cooked, right? So everyone who happened to order the chicken got sick that night. So it had nothing to do with passing anything between people. It had to do with a common origin. And there are various epidemics in history that are shown to occur this way. And one of them is yellow fever. So back in revolutionary times in the United States, there was a major outbreak of yellow fever in Philadelphia. And I think even George Washington had to flee the area in order to avoid being sick until everything blew over. And they were able to figure out what caused that outbreak, and it was related to a bunch of dead animals and sewage that was floating in the water that supplied the drinking water. Oh, and wow. What happened is that, like I said earlier, as animals decompose, the microorganisms that recycle them make some compounds, just like the endotoxins I described in that experiment, that are quite toxic and can cause disease. So they were basically drinking these toxins in the water, and it made everyone sick with yellow fever. Once they removed this material from the water, everyone got better. So, and a lot of people were sick, and I'm sure some of them thought that maybe they were passing it back and forth between each other at that time as well.
So what about things like venereal disease?
Yes. So, well, a lot of people have actually asked me about that, and they've asked me specifically about the so-called herpes simplex virus. And I think this is a great example to discuss because it is something that is a real nuisance, and it causes people embarrassment and shame. And I've actually heard about some people who were able to get better from this by some unconventional means that I can mention if you like.
Sure, absolutely.
But people are definitely have a lot of misunderstanding about HSV. I'll just refer to it as that. And once again, with HSV, the proof that there's a virus that causes this outbreak illness has once again not been satisfied. But what the thing is that people don't realize is if we did these antibody blood tests, which is how they say that you're HSV positive or not, that over 90% of the population is actually positive for these things. So it has nothing to do with sexual transmission. It's, that everybody has these positive tests, and there's really no clear experimental evidence of how whatever it is that causes these tests is in all of the people that they test. So I don't really know the answer to that, but if over 90% of the population has it, it doesn't really fit the pattern for a sexually transmitted disease. But the thing is that even though almost all of us, like even if you and I, Jimmy, got tested, we would most likely be positive, but we don't have the outbreaks. And the outbreaks of, the blisters and cold sores around the mouth or on the genitals only happens in a small portion of people. And so why would we blame this illness on a disease that only a tiny fraction of the people who supposedly have the virus get the symptoms of it? What about all the healthy people who test positive for it, right? So you could see that it doesn't meet the criteria for causing a disease. So in most of these infectious diseases, like this herpes simplex disease and things like chickenpox and measles as well, is that the skin is a place that can help us by removing toxic substances from our body. And when certain conditions arise that perhaps we get overloaded with toxins, or perhaps there is a weakness in our skin from some kind of nutritional deficiency, that our bodies create an eruption. And that's how they basically get the toxic material out. And this may happen when there's not another convenient route out of the body, but it's really related to that. And so the same thing, why would people who congregate together get this illness? Well, because they're all exposed to the same toxins.
Sure. What I'm getting from a lot of this, and from what you're saying and from what a lot of other people are saying, is these substances called germs or viruses or fungus or whatever are the body's response to being poisoned by one thing or another, whether it's a chemical poisoning, a electromagnetic poisoning. But it seems like that's what's going on. Am I off track by saying that?
No, not at all. You did leave out a physical injury as well.
Right.
But in recent years, we have come to learn a lot more about the microorganisms in our body. Mm-hmm. And I think people are now very familiar with what's called the microbiome, or the gut flora or gut microbes. Like those are some common words, right? And now it's known, and I can't believe that this wasn't known 100 years ago when they were looking under a microscope at all these things. But now it's commonly known that actually these microbial cells outnumber our own human cells in our body, in some papers by a factor of 10 to 1. So we're actually mostly made up of microorganisms. If you want to look at a numbers game, of course, the human cells are substantially bigger, so they weigh more and they take up more mass. But in sheer numbers of cells, we're basically walking sacks of microorganisms. So
yeah, in a sea, in a very poisonous environment in our world, right?
Yeah, absolutely. And let me tell you further that if you destroy these microorganisms that are living in your body by various means, it does not sustain life. I mean, earlier in my career, I had some experiences in cancer medicine, and many times people, due to their illness, were unable to eat normally or take food by mouth, like especially if they had throat cancer or cancer of the stomach, and their whole gastroenteric tract was not functional. So they would sometimes give nutrition through an IV. They call it TPN, or total parenteral nutrition. And there were many cases when I thought, being somewhat naive and very early in my career, that, well, wouldn't this be a simple solution for this person that we just feed them, IV nutrition? And the doctors with experience would always say, no, we don't want to do that. Let's wait. That's the last resort. And I was always confused because it seemed so convenient, and why wouldn't it work? And then I did see that sometimes when this approach was used, the person would just do horribly. And it wasn't until 10 or 15 years later that I realized, oh my gosh, the reason they do so poorly is because all of the microorganisms in their gut are starving, because they bypass feeding them. Because when we eat, we're not just feeding our human cells, we're feeding all those bacteria. And this is why, people tell you to eat high fiber, that it makes your colon work better. And it's because it makes those bacteria happy. And so they function like they're supposed to in harmony with our body and allow you to digest food and excrete the toxins.
That makes so much sense. Why can't we get that information out? I really, and this is my opinion, I really feel like we've been poisoned from every angle. We've got people spraying chemtrails in the sky. We've got glyphosate in our food supply. We've got fluoride in the water. It's amazing that there's a civilization still alive, but this is sort of the slow kill method as far as I'm concerned. I know that's a sort of a pessimistic way of looking at it, but we have to be realistic about what's happening in our world, and we've got to take a stand at some point as human beings if we want to continue and not only be healthy but to have our freedoms, because at this point all of that is in dire jeopardy. So you're listening to Inside the Matrix with our special guest, Dr. Andy Kaufman. We got one more segment to go, so stick around with us. We'll be back in just a few minutes, and if you have a question, pop it in the chat. We'll see you in a little bit. All right, welcome back, everybody, to our final segment of Inside the Matrix today with our special guest, Dr. Andrew Kaufman. Now, I know there's been a lot of hysteria being promoted in the mainstream media about all these people testing positive. Oh, we got celebrities, we've got politicians, we've got, Bozo the Clown. Everybody out there is testing positive for, coronavirus. So what are these tests that they're using, Andy? And are they, in fact, effective in any way? Or what are they testing for? Give us some insight on that.
Well, I'd be happy to. And I just want to say that there's a couple of important points, that none of the tests are testing for a virus because they haven't identified and purified any virus. And they didn't, so there's no test that tests for the virus. So when they're testing your blood or other tissues, they are not finding a virus or even looking for a virus. What they're looking for in the original test, which is called a PCR test, and that's a way to amplify or increase the numbers of basically a needle in a haystack so that there's enough of it that they can identify, is testing that genetic material that I mentioned. But as I described before, there's no certainty as to the source of that genetic material. So even if you could show that it was present, it really doesn't tell you much information since you don't know where it comes from or what it means. And the test for a PCR has never been compared to what's called a gold standard. And all new diagnostic tests have to be compared to a gold standard to show that they are accurate at all. And if you do that, you can calculate an error rate. And if that error rate is small enough, like maybe 1%, then you say, well, this is a pretty good test. But if you have nothing to compare it to, then you have no known error rate. And basically, you have a test that doesn't actually show anything.
Imagine that.
So when there was a paper that tried to estimate an error rate from this test, and they couldn't compare it to a gold standard, but what they could do is they could test people and see whether they were positive or negative, and then they could follow those people out to see if they were sick or not. Okay, so it's not very accurate, it's an estimation, but it was the only real thing they could do because they didn't have the gold standard. And they estimated that the false positive rate of this test was 80%. So that means that 4 out of 5 people who had a positive test did not get sick.
Mm-hmm.
So what we're doing really is labeling a lot of people as positive for something that has never been proven to exist or cause illness, and then putting these numbers that seem really high to people because people don't really know how many people are sick and die in a given year. No. But it sounds like a heck of a lot of people because, you have to really do math. You have to divide it by the population. right? So you have to divide it by 300 million, and no one's looking at that to see that actually, oh, it's not that many, or compare it to how many people, get the flu in a given year, or how many people die from traffic accidents or from smoking, right? All of these kinds of issues, to get a sense of what that number means. It just sounds like a really big number. Like if it were dollar bills, you'd say, oh, that would be a lot of dollars, right? But essentially, it's completely meaningless because what is the testing for? We don't really even know. But it certainly creates a lot of fear. And then they just released a new test. And you know that the companies that are making these tests and rushing to make new tests, this is a huge, huge windfall for them because whoever wins the race with the test that they choose is going to be sold all over the world to everyone instantly. So it's going to be billions and billions of dollars for that company. So they're trying really, really hard, just like there's a race to come up with a vaccine right now. There are companies all over the world busting their butt, probably working day and night to come up with something because they want a piece of that pie. So when there is an urgent situation and tests and drugs and things suddenly get invented, the government, who's supposed to regulate this and make sure that these things are safe and accurate, they often just rush and give permission. And so the FDA gave permission for a new test that is an antibody test. They call it a rapid test because the PCR was kind of frustrating because it took several days to get results. So now there's this new one that's rapid. It can get results within a couple of hours. But the thing is that it's not FDA approved, right? Now, we all know that diagnostic tests and therapeutics have to undergo this approval process where they have to have, not just one study, they have to have actually 3 phases of studies to get approved if they're a therapeutic, and they have to have specific criteria if they're a diagnostic that is much more extensive than one single study. So, and there's a process that takes time and it has to be reviewed and decided upon very carefully. And they would not approve a test, generally speaking, that had no gold standard to compare it to. But in this case, they didn't approve it. They authorized it, which basically means say that it's okay to sell it. You're not going to get in trouble. But it doesn't mean that we say that it's effective or accurate. And in this document where it's like I'm not sure if it's a press release or not, but it basically gives all the information. No, it's actually a letter. I think it's the authorization letter to the person at the company who made it is what it is. And it says here in section 1, criteria for issuance of authorization. I'm gonna read this because it is very, very telling. Okay. So I'm going to read bullet point 2, and I think everybody can find this document if they want to look at it themselves. And it says, I quote, based on the totality of scientific evidence available to FDA, it is reasonable to believe that your product may be effective in diagnosing COVID-19. So they use not 1, not 2, but 3 words to add uncertainty into this claim. So they say it's reasonable, right? Now, and that they believe that the product, they don't know, right? They believe, not know, and that it may be effective. So and it may also not be effective in diagnosing COVID-19. So they're essentially right here admitting that this test doesn't measure anything that they can say for sure what that is. So this is just further misinformation and obfuscation. And I'll bet that there is a lot of positive tests when they start rolling this out.
Oh, you bet. They got to feed into that hysteria, in order to keep this game going. Now, the other aspect of this, too, is that you've got all these companies now getting these billions of dollars to, like Bill Gates and that cluster of demons that inhabits his body, sponsoring these companies to come up with a vaccine that is going to further poison people. So, in your opinion, Has any vaccine that you are aware of, and I'm sure you've read a lot of scientific studies, been either safe or effective?
Well, this is a very important thing to discuss. And, for most of my career, I just was acting under the assumption that vaccines were safe and effective because that's what I was told during my medical education. And the emphasis there was really not on demonstrating that vaccines are effective. It was more about learning which one to give at what age. Because that has become so complicated because I believe now there's something like over 30 different vaccines and they're given multiple times. And there's, you need like charts and tables just to figure that out. So that's what the emphasis on. Certainly, they told us the theoretical basis that you expose a person to some kind of infectious agent and your immune system reacts to it and creates antibodies. And those antibodies stay around and confer immunity so that if you are challenged with that infectious agent later down the road, that it doesn't make you sick because you're immune to it. And that sounds like a pretty darn good story. So I took it at face value and learned the schedule. And when I was on my pediatrics rotation, I did what I was told. But later on, when I started looking into the basis of allopathic medicine, and at the same time I started learning about other methods of healing from a variety of sources, this is just an area I'm fascinated about and want to study as much as possible and learn as much as possible. I started to investigate the research about vaccines, and I came across some astonishing findings. So you can find this historical data that goes back, more than 100 years, and they were collecting statistics basically about how many people in the country. And this is what the CDC does this is what's called public health and epidemiology. It's a science. It's not the most accurate science, but it's very important in order for us to know what's going on with our health in case things change or something comes along that makes more people sick. We want to be able to recognize it. So these illnesses that were very prevalent in the past, like measles and diphtheria, polio. And polio, absolutely, that's a great example. So they were monitoring these very carefully and hoping that they would go away at some point. And so you can find these graphs where it shows, 1910, 1911, all the way up to the time when these illnesses were not prevalent anymore. And, nowadays we don't know anyone with measles practically. It's pretty rare. There's a little outbreak here and there, but, almost never does anyone die from it. It's a pretty harmless disease. Only people who are already very sick might succumb to any serious complications. And certainly we don't know about people having polio all the time like they did in the past. And so all of these diseases decreased. And we're all told that the reason these diseases went away is because of the vaccines. But when you actually look at these graphs and you see it start to slope down and get all the way down right to where it is currently, and then you put a little arrow on that graph of the date that the vaccine was rolled out and given to people, you see that occurred after the amount of cases already went down practically to zero. So if these diseases went away before there was even a vaccine available, it certainly doesn't take much scientific scrutiny to realize that the vaccine couldn't have possibly caused the reduction in the disease. So every vaccine-related illness that I could find this data on showed the same exact pattern. So there was really no evidence whatsoever that the vaccines did anything to prevent these diseases. Now, I want to take this one step further because this is just such an important issue, which is that there is a standard way to evaluate therapeutics like vaccines or drugs or surgical procedures or other procedures that are for therapy purposes to prevent or cure or improve disease. And that is called a placebo-controlled trial. And this is really important because the placebo and what it really means to our health has really been lost in our modern society. That we don't realize we actually have quite a lot of power just with our mental fortitude to actually relieve ourselves from illness. And this is so powerful that the medical establishment actually tries to piggyback on this placebo effect by doing a variety of things. Like, this is probably one of the main reasons for doctors wearing a white coat, because that white coat itself actually makes you feel better as soon as you see someone in it. You know, except for the small number of people who get really nervous around it. Right. But for most people, but even with the pills, for example, like, it's been shown in several studies that if you can have just a plain sugar pill, and if it's a tablet or a capsule, the capsule is actually much more effective than the tablet. Now, you take that a step further. If you add color to it, the colored pill is more effective than the white pill. And you can take this yet another step further. If there is a brand name or a number printed or embossed into the tablet, then it becomes more effective than one that is just plain. So these all have to do with our belief in, is this thing going to help us? And those things increase our belief because obviously someone put more time and thought into it, and it's more pleasing to the eye, so we believe in it more, and thus we get better more. So in order to compare if the actual chemical in that tablet really does anything, what we have to do is make two tablets that look identical, and one contains the actual chemical that they say is a medicine that they're testing, and the other one contains something that couldn't possibly do anything to our body, like sugar. I mean, sugar does something to our body, but in that minuscule amount, it's not going to make us recover from a disease. And then you can look at the improvement in the placebo group and the treatment group, and you subtract out the placebo effect, and that's what the chemical drug actually does. Right? And you need to do that because if the placebo and the chemical drug have the same benefit, then the drug actually has no benefit whatsoever. So if you're going to do this with a vaccine, the way you would do it is you would have to do the same exact procedure. So you'd have to have one syringe that was full of the whatever they're putting in vaccines, and I don't think we even really know exactly what they put in there, but I can certainly tell you about the things that they tell us they put in there. And then you'd have to have the other group would have to have a syringe with just salt water in it, because we know salt water is not going to prevent any diseases. But if you don't give the injection to the placebo group, one thing is they know they didn't get anything. So they're not going to have that placebo effect. And you know there is a big placebo effect from an injection because people think that injections are more powerful than taking something by mouth, even though that's actually not true at all, not even one bit. It only has to do with the amount of the drug that you actually take. If the amount is equivalent, there's no difference between a pill and an injection except the placebo difference, because you believe the injection or the intravenous medicine is superior, but it's actually not at all. In fact, it's more dangerous because every time you put a needle into the body, you're introducing potentially toxic substances into the body directly. You know, the skin is a very important barrier. In fact, it's our biggest organ of defense that keeps, poisons and bad stuff out of our body. So we don't want to break that. So this standard of having a placebo-controlled trial is the gold standard for the FDA to approve any therapeutic. If you don't have a placebo-controlled trial, don't even bother putting in the application because it's going to be an immediate no, except for one thing, and that's vaccines.
Ha! Wonder why.
Well, they made this very strange argument saying that since vaccines have been proven to be so effective, it would be unethical to do a placebo trial because then the placebo group wouldn't get the benefit of the vaccines. The only problem is they never proved the benefit of the vaccines in the first place. So actually, what they're saying is it's much more dangerous now because you're giving people something that hasn't been proven to be effective. So,
Or safe.
Well, I'm going to get to that in just a second.
Okay.
But so you have no historical epidemiologic data that shows any evidence whatsoever that vaccines are effective. And then you have no placebo-controlled trials to prove that they're effective. So in aggregate, there's actually no proof that they're effective. So I'll say that if it's up to you, the decision of whether you would accept this or give it to your child, you don't need to go any further at all. Because why would you even consider taking it if it's not going to do any beneficial thing for you? Now, I will say that it has immense potential to cause harm because these vaccines have a lot of things in it that we all know are toxic, right? Like aluminum, or specifically aluminum hydroxide in a microparticulate form. Mm-hmm. And, we all know that aluminum is poison. And why would you want to put this in your body? Now, I know that a lot of government officials might say that, well, it's not actually, it's a tiny, tiny amount. And if you assume that it's distributed evenly throughout your body, that it wouldn't be in a high enough concentration in any particular part of your body to cause any problems. And that sounds really nice, but it's not based on actually studying what happens when you put aluminum into the body. And that science has only been recently been done to any degree over the past 5 or so years. And it's been extremely enlightening because what it's shown is that when you put this aluminum into the body, that it doesn't distribute evenly to every part of your body and dilute itself. In fact, it actually stays right at the injection site because your body, when you give the injection, you're breaking the skin and causing trauma. So your various cells from your immune system go right there to the wound because that's what they're for, to protect against invaders, right? And that syringe is the invader. Right. And then they get there and they're like, oh my God, what is this metal stuff? Right? Because in nature, aluminum is not really present in the soil or in the plants like other minerals like arsenic. So it's a result of industrial mining, and the body doesn't really recognize it or know what to do with it. So what it does is they have these big cells that gobble stuff up. They call them macrophages. They gobble up the aluminum, and then they create this walled off, like secluded area. It's really a quarantine in your body. They call them granulomas, and it contains the aluminum all at that one site. And this protects your body from the toxicity. But the problem is that it doesn't ever go away because the body has no mechanism to break down aluminum. Like antioxidants don't break down aluminum, enzymes don't break down aluminum. It's an element and it's stuck there. So at some point, the immune cells are going to abandon that granuloma and they're going to go somewhere else and they're going to take the aluminum with it. And they're taking that whole shot of aluminum with it. So remember, it doesn't spread out to small, tiny amounts. It's that one big dose. And if those cells go to your brain, then it could cause various types of neurologic problems. And that's right. Often what we see.
Right, like dementia, Alzheimer's, that type of thing, right?
Well, also Guillain-Barré syndrome, which happens from the flu shot and many other vaccines where you get paralyzed, sometimes, often permanently.
It's terrible. And we're coming close to the end of the show, and I want to thank you so much for sharing your knowledge with us because people need to know that they are being lied to in a gross and bizarre way at this point by our own government. And we haven't even touched on other aspects of this phenomenon that we're seeing and that the dangers of the radiation coming from 5G, and the symptoms are very similar to what they're talking about with this supposed coronavirus. And the fact that this 5G has been rolled out in Wuhan, China, in Italy, in all these other places that they say that they have these, tremendous outbreaks. So that's a big question mark, and there's a lot of good information coming out. So please pay attention to that, people. Radiation poisoning could very well play into a lot of this. So we've only got a few seconds left. Again, Dr. Kaufman, thank you so much. Keep up the good fight. We so appreciate and respect people like you that are risking, your professions to tell the people the truth. And in my opinion, you are a hero, and we thank you. So anybody, stay safe out there. You've been listening to Inside the Matrix. We'll see you again next week. Thank you.
This is Inside the Matrix, hosted by Jimmy Brent.
Who are we talking about?
Jimmy Brent. Live every Sunday, 3 p.m. Pacific, 6 p.m. Eastern. Exclusively on the KCOR Digital Radio Network.
It is worldwide.
Broadcasting from an undisclosed location deep within the neon capital of the world, Las Vegas, Nevada. For more information on the host and the show, please visit InsideTheMatrix.org. That's InsideTheMatrix.org. It's time to break the chains of manipulated consciousness and claim our true nature as creative, divine beings. Inside the Matrix. Inside the Matrix.
This is KCOR Las Vegas, home of the Digital Radio Network, broadcasting from a shack just south of Area 51.
Wait, that doesn't exist.
This is the KCOR Digital Radio Network. Now for the news.